right, so today we are starting the information for first information for the next test. And it is the beginning of the unit that talks about the target theory and cell survival curves. In your textbooks, this information is on pages 128 to 132 in the radiation protection textbook, which is the Statkowitz-Shearer text, and it's on page 761 in the Carroll text. So that's where you'll find the reading. And again, it's not much reading, it's only like 4 pages. So we've mentioned the target theory before in the very first lecture of the semester, and we've mentioned the term targets. So we've also talked about energy exchange from radiation. Radiation is administered to a patient. When that radiation interacts with the patient, it's interacting randomly, and it's interacting with the different atoms and molecules in the patient's body. It's most likely to interact with water molecules because there are more water molecules than anything else. But the energy can be transferred within the structure or within the cell until something important is damaged. So those important or critical sites within a cell are referred to as the targets. And again, there can be numerous targets. A radiation biologist could identify the mitochondria as a target. Another radiation biologist might identify the ribosomes as a target. Another radiation biologist might select the DNA as a target. But targets in general are sites within the cell that carry out very specific functions. If the target is damaged by radiation, what that typically means is that the function of the target is lost. So if you're studying the mitochondria, the powerhouse of the cell producing energy for the cell, if the target is damaged, you would have decreased energy production or no energy production. If you selected the ribosome, if the ribosome is damaged, maybe you get decreased or impaired protein synthesis. So again, targets are identified as very specific sites within a cell. They have very specific functions. If they are damaged by radiation, their normal function may be impaired. And if their function is impaired, then the cell could potentially die. So the target theory says that when we irradiate, energy is transferred into the cell. It can be transferred either via direct action or indirect action. Energy can be exchanged via ionization or excitation. We know that's how direct and indirect action works. But again, if the energy is transferred to the target and the target is damaged, then the cell may cease to function. We use a term to describe the radiation damage to a target. And the term that we use is a hit. We will say when there's a hit on the target, the target is damaged. So when we use that term, it means that there's unrepaired damage to the target and the target is not functioning properly. Even though we can determine if our target has been damaged because it won't be functioning properly, there's really no way to determine exactly how the damage was done. We won't know if the damage resulted from an ionization or an excitation. We won't know if the damage occurred directly or indirectly. But what we will know is that ultimately energy was transferred to the target and the target has lost function. As we mentioned earlier in the semester, the target theory was developed over a number of years by a group of scientists, Speer, Leah, Zimmer, Timofov, Rozovsky. I have no idea if that was spoken correctly, but that's my guess. So they were the scientists that developed the target theory. And for most research studies, the primary target is DNA or the chromosomes. We mentioned just a few minutes ago that other radiation biologists or that radiation biologists might be able to pick a variety of targets, the ribosomes, the mitochondria. They typically do not, and the reason that they don't is that those organelles have redundancy. So there's multiple mitochondria in a cell, there's multiple ribosomes in a cell. So even if some are damaged, the cell may still be able to function properly. You really only have one set of DNA and one set of chromosomes. So when they're damaged, it's just easier to pinpoint them as the target. So for all purposes, when we talk about the target in this unit, we're going to talk about DNA. DNA is our selected target. So the target theory has been used to study radiation effects on cells. And the most common effect that is studied is cell survival. And cell survival is defined as the cell's ability to reproduce. Can it successfully go through multiple cell divisions? If we were doing an experiment and we irradiated all these cells, if the cell population could not reproduce, we would say that it was killed, because by definition, we kill it when it can no longer reproduce. Because if a cell line fails to reproduce, that means that that cell line will become extinct. And you may say, well, what about the cell lines that don't reproduce? Nerve cells that never reproduce, muscle cells that only reproduce in certain situations. If you were looking at those specific cell types, you would have to look at a loss of a specific function. But for all other cell types that are studied, typically the end point that's studied is cell survival or the ability to reproduce. So before we look at cell survival curves in depth, we want to talk for just a minute about general cellular responses to radiation. And so this information, the best information is probably in the Statkowitz-Shearer book, and it's on pages 131 and 132. So we're going to talk about 5 different cellular responses. We're going to talk about mitotic delay, mitotic death, reproductive death, apoptosis, and then what's called instant death. None of these sound particularly good, and a lot of them sound very similar. All right, mitotic delay. So what is mitosis? All right, somebody tell me, what is mitosis?
Cell division.
Cell division.
It's cell division. Okay, somatic cell division. So mitosis is cell division. If a group of cells is irradiated, they may experience mitotic delay. Well, that's pretty self-explanatory. Mitotic delay simply means that the cells don't divide at the right time or when they should. So how do you know when cells should divide? How do you know?
Depending on the cell, like the red blood cell lasts 120 days, right?
Like 120 days. Kiara, you're exactly right.
Versus the white blood cell might last, I forgot how long, but it's longer or shorter.
Shorter, much shorter. All right.
Like everything has its own time period.
Everything has its own biological clock. Excellent. So Kiara's exactly right. Red blood cells have a lifespan of like 120 days. So they're going to divide about every 120 days. White blood cells is much shorter, only like 3 to 5 days. So they're going to divide every 3 to 5 days. Skin cells, your epithelial cells of the skin, about every 3 weeks. So cells divide depending on the cell type and what is their biological clock. But if you irradiate cells, some of the cells in the population won't divide on time. If we look at this chart, here we see a graph of basically the mitotic index of a cell population. And the mitotic index is the ratio of the number of cells in division at any one time to the total number of cells in the population. So if we say that a particular group of cells in your body has a mitotic index of 0.25, what that means is 25% of the population is dividing at any one time. And the mitotic index should be stable. So what we see here is that this line is flat. Whatever it is for this particular cell population, it's stable until all of a sudden we irradiate the population. Look at what happens to the mitotic index. After irradiation, we see the mitotic index is no longer stable, but it drops. And then over time, the population recovers. And then it's too high. So our 25% of cells dividing may drop down to 10%, and then it may go up to 40% as the population recovers. So we know that irradiation disturbs the mitotic index. So how much radiation does it take? Well, it depends. It's very variable. Anywhere from 0.01 gray or 1/100 of a gray all the way up to a full gray. So it's variable with cell type. But when it happens, the cells are typically delayed in early S phase or in G2. Oh my goodness, that means we have to remember the cell cycle. So what are the stages of the cell cycle?
S phase, G1, G2, and it's one more.
Uh-huh. What's it called when it's
Uh,
dividing?
the the telophase? No,
Nope.
it's not. It's
You're
it's
thinking
the
of
M,
it's
S,
simply
the
M
S,
phase.
S2. Oh, M
M.
phase. That's what
M
it is.
phase.
M is the only one he didn't say.
All right. So when we think about the cell cycle, M phase is cell division and has 4 stages: prophase, metaphase, anaphase, telophase, but all of that lumps under M phase. Then we have G1, which is the first gap or the first growth phase. Then we have S phase. What happens in S phase? I couldn't, whoever said something, I couldn't hear you.
Interphase.
It is part of interphase, but what specifically is happening in S phase?
It's a synthesis phase.
It is the synthesis,
Is it the
DNA
DNA
synthesis, so the
replication?
DNA replication. And then we get to G2, which is the second growth or the second gap phase, and we have RNA synthesis, protein synthesis. So when mitotic delay occurs, for some reason, cells get stuck in either early S phase or G2. They're not exactly sure what happens, but they've theorized that some of the chemicals involved in cell division are altered. It could be that protein synthesis is slowed down, so some of the proteins are not ready, or it could simply slow down the rate of DNA replication. But again, they're not exactly sure, but all of those things are theorized as potential causes for mitotic delay. So when cells are irradiated, some of their cells, when a patient's irradiated, some of their cells may not divide on time. And again, the dose range is 0.01 gray, or 1/100th of a gray, up to 1 gray. So my question is, are there any common procedures that you can think of that would give a dose to the patient in this range?
The highest,
Or do you have any idea?
Nuclear medicine?
Nuclear medicine might, in a particular, if it was like a therapeutic dose. But think about general diagnostic. Don't go to nuclear medicine. Think about general diagnostic.
I was just thinking the highest doses are in Flora, right?
Mm-hmm. Potentially. All right.
Myelogram.
A myelogram. Myelograms probably aren't too bad unless they take a long time. If you have your Statkowitz-Shear textbook, open it to page 32. Because we're going to look at a chart that gives estimated doses from diagnostic exams. So tell me, what is the entrance skin exposure from a chest x-ray?
0.1 milligray.
0.1 milligray. Okay, 0.1 milligray. What is that equal to in terms of gray? If you do the math, millis 1/1000th, a chest x-ray would be 0.0001 gray. So would a chest x-ray likely cause mitotic delay?
No.
No, because it's much lower than the 0.01 gray by 2 powers of 10. Well, let's look at an abdomen. What's the entrance skin exposure for an abdomen?
That one is 4, so
It's
I
4
guess it-
milligray or 0.004 gray. Still below the limit. Everything on that chart, from a skull x-ray to a pelvis or extremity x-ray, is below the threshold for mitotic delay. So none of the exams that we do in general diagnostic radiology should result in mitotic delay. Now, for fluoroscopy, I was actually looking for a table, see if I could find it in the book, in your book, about fluoroscopy doses, because yes, they are higher. And let's see. See if I can find. Well, I'm not finding any listed in this textbook, but just know that potentially a long fluoro study probably could. It could reach that threshold of 0.01 gray. That would be, it would be a relatively long case, and so we're probably not seeing very many of those. But anyway, mitotic delay, not likely to be caused by any general diagnostic procedure, potentially by a fluoroscopic procedure. Like I said, here are the potential causes: altered chemicals and mitosis, not having the proteins that you need, decreased rate of DNA synthesis since it gets stuck in S. And here is your cell cycle as a review. You know, G1, cell growth, S, DNA replication, G2, more cell growth, and then M phase, which is mitosis. All right, so the next thing that can happen to cells after irradiation is called mitotic death, or mitotic death, or genetic death. And this occurs or can occur at relatively low dose levels. So it is possible that a general diagnostic study could cause mitotic death or genetic death. When this happens, basically the cell will attempt to divide, and it may be able to divide once or twice, but ultimately genetic mutations that were caused by the radiation will inhibit further cell division. So mitotic death or genetic death ultimately will kill the cell. It may be able to divide once or twice, but it's not going to be able to divide continually. So ultimately the cell line will die. And again, the culprit are those genetic mutations that have occurred because of the irradiation. Since mutations are can occur at any dose level, no matter how small, low dose levels can result in mitotic death. Next, we look at reproductive death. And you're going to think that this sounds exactly the same. And it is very similar. In reproductive death, your dose range is higher, somewhere between 1 and 10 gray. The cell may be able to reproduce or go through one cell division, but ultimately the daughter cells are not viable. The daughter cells die, so the cell line is finished. But before it tries to divide, the cell will function as normal. It's still going to have normal metabolism. It's going to have normal protein synthesis. Everything was going to be normal, but when it divides, for some reason, the daughter cells are not viable. In the dose range of 1 to 10 gray, this is outside most any general diagnostic procedure. So again, this is not something that we would be concerned about in general diagnostic exams. Because remember, we are administering very low doses of radiation, relatively speaking. The next effect on the cell in general is called apoptosis. And apoptosis has had numerous names over the years: non-mitotic death, non-division death, programmed cell death, and interphase death. You will still hear the terms programmed cell death and interphase death associated, or you'll still see those in the literature.
Why so many names?
I have no idea. To confuse all of us, basically, I think.
Oh, okay.
Maybe different scientists came
Well,
up with it
worked.
at the same time and just called it different things. That's typically what happens. Scientists all over the world come up with the same concept and they all name it something differently and publish at about the same time. But anyway, this is sort of an interesting phenomenon. But with apoptosis, the cell dies without ever attempting to divide. Now, with reproductive death and mitotic death, the cell divides. You know, with mitotic death, it may divide once or twice and then it just can't. And with reproductive death, it divides once and then the daughter cells die off, so it can't go any further because the line is killed. But with apoptosis, just the cell never even attempts to divide because the cells basically just fall apart. So if you see this diagram, it goes through the stages of what happens in apoptosis. And this you'd have a normal cell. After radiation, the cell first starts to shrink, so it's condensing. Then it starts producing these, they're called blebs, and it's, let me see if I can find the thing. They start to project outward. So look at the cell membrane. It's not smooth anymore. It's very irregular with all of these projections outward. After that, the nucleus totally breaks down and breaks apart. Ultimately, the entire cell breaks apart into fragments, but the organelles themselves don't break up. It's just bits of the cell and whatever organelles are in there just exist. And then, of course, because the cell essentially is dead, it's now trash, so the fragments will be cleaned up by the cell's cleanup crew. One thing that's interesting about this process is that it is a normal process that occurs in certain organisms. In humans, when the fetus is developing, the hands, the fingers and the toes are webbed. But as the fetus develops, apoptosis occurs and the webbing disappears. So that's normal. Why does it happen? Why does apoptosis happen? They think also it's biochemical changes, but they're not exactly sure what causes it. You've probably figured this out. There's still a lot of uncertainty in radiation biology.
Hey, Dawn.
Okay.
Could apoptosis be described as like a specific form of phagocytosis?
I don't know. I've never really thought about it that way. I mean, ultimately the cell is digested, but it's not until it all breaks apart. So it's not like bacteria where your body's cells just go out and, you know, envelop the bacteria and they eat it that way. But ultimately it is digested by the cell because it's broken up and it's trash. I don't know. I'll see if I can find something on that, because no, I've never really thought about it that way. But ultimately it is gobbled up. One thing to note, look at the dose range. The dose range for apoptosis is very broad, from as little as 0.5 to 1 gray, all the way up till several hundred gray. And it is dependent again on cell type, on the radiosensitivity of the cell. Obviously, it would take a larger dose for a highly differentiated, non-dividing cell population. Since that's the case, whose law is it following? Who developed the law about sensitivity of tissues that said they were dependent on differentiation and mitotic rate?
Those two French guys.
Becquerel,
the two French guys.
isn't it Becquerel and Lawrence, something like
It's
that?
not Becquerel, it's Bergenet
Bergenet.
and
sorry.
Tribundeau. It's the law of B and T. Becquerel was the scientist dealing with radioactivity, but at least he was a B. So the law of B and T says that cell types that are highly differentiated and slowly dividing are more resistant. So it would take a higher dose to cause apoptosis in those populations than it would in a simple, rapidly dividing cell population. So again, when we think about effects to the cell, we can have mitotic delay, where cells just don't divide on time. We can have mitotic death that is due to genetic mutations. We can have reproductive death, where basically the daughter cells are not viable. And then we can have apoptosis, where the cell basically just falls apart. The last of the effects on cells is instant death.
Woo!
Instant death sounds really bad, because it is.
It sounds really quick.
It is really quick, because look at the dose level. This occurs at doses greater than 1,000 gray. If you administered 1,000 gray to a person all at once, that person's dead, you know? So think about it on the cellular level. Doses that high literally cause the DNA to fall apart, the proteins congeal like Jell-O, and the cell membrane ruptures, and literally the cell is destroyed. But that is a very, very large amount of radiation. Nothing we do, even in radiation therapy, would give that high a dose acutely. Radiation
So
therapy
that would be,
exceed that dose, but over, say, a 30-day or a 40-day time period, not instantly. So instant death occurs at extremely high doses because literally the cell just,
because you nuking it. You basically
breaks apart.
nuking it, right? Or
well, basically, yes.
I have a question, Don,
Yes.
about instant. Like when people get radiation poisoning or sickness,
Uh-huh.
would this go into this where they end up dying from it?
It could, and it would depend on the source of the radiation and how much radiation they received. You know, for example, if you were close to the Hiroshima or the Nagasaki bombing, not close enough to be obliterated by the bomb and the heat, but close enough that you received instantaneously a very high dose of radiation, yes, that could have resulted in instant cell death. And remember, we're talking about on the cellular level. The doses, very high doses to a human being, would cause an acute radiation syndrome. And we'll talk about those a little bit later in the semester. Some of them can be fatal in as little as 24 hours. But even high dose radiation doesn't kill you like immediately. It takes a little bit of time. Not much, but a little bit. Okay, so again, when we think about cell death, there's multiple times, depending on what exactly happens. And as we looked at them in terms of increasing dose. So the highest dose, greater than 1,000 gray, can cause instant death. After that, or before that, in terms of dose level, would be apoptosis, because it's very variable depending on cell type. Then there's reproductive death, 1 to 10 gray, mitotic death that can occur at very low dose levels. And then mitotic delay, which is also a variable, but somewhere between 0.01 and 1 gray. So all of those are different things that can happen at the cellular level when we irradiate. All right, so it's 2:30. Let's take it, let's stop for a second. Let's take like a 5-minute break. And we will come back and go into the second part of this lecture.
Can we just stay?
Okay, so we have been looking at the effects of irradiation on cells, and we had talked previously about the target theory. So now we're going to look at how the target theory is applied and how radiation biologists, cell biologists, physicists can all work together to try to determine how different cell types respond to irradiation. So when we are doing what we call cell survival experiments, what we're trying to determine is, can the cells reproduce after irradiation? So you're going to start with basic laboratory equipment. You're going to have a certain cell line or cell type, and you're going to have some medium in which you're going to plate your cells. And you probably all did this in either high school science classes or college science classes, where you literally would plate cells. And in this example that's illustrated, they've plated 6 different cells, and then they incubate them. So you incubate them maybe 24 hours, 48 hours, and post incubation, you hope to see groups of cells. And those groups of cells we would refer to as our cell colonies. Normally, cell colonies should grow because there's nothing inhibiting their growth. So in this particular example, they plated 6 cells, incubated the cells, and they had 6 cell colonies. So these cells were perfectly able to reproduce, which you would expect because we've not done anything to them. So if we are going to do cell survival experiments, we're going to do something a little bit different. We're going to add in the radiation to see how that influences cell survival. So you would plate the cells, whatever cell type you're studying. You would irradiate those cells that you had plated to different levels of radiation, so different dose levels. Then you would incubate them. After incubation had occurred, then you would check for cell colonies. If you plated 6 cells and after irradiation you had 6 colonies, you have 100% survival. If you plated 6 cells and after irradiation you only had 3 cell colonies, then you have 50% cell survival. So it's pretty simple math. In reality, when these experiments would be done, you would be dealing with multiple plates of cells. You would have many different cells plated. You would irradiate a certain number of those plated cells to each dose level to ensure that something else wasn't happening, okay? So there'd be a lot of redundancy, but you'd have a lot of cells that you would be dealing with. But it's a pretty simple concept. If the cells cannot reproduce and produce cell colonies, then that dose level of radiation killed the cell. Remember, our definition of killing the cell means that the cell cannot divide. So over time, many different cell types have been studied with cell survival experiments. And the experimental results are always displayed graphically. So you have a graph of cell survival. In the cell survival graphs, the x-axis is always the dose axis. So we have 0 dose up to a maximum dose level, always on the x-axis. The y-axis is always surviving fraction. So for every cell survival curve that you have, and you can look at all of the ones that I handed out to you in the handout, x-axis is always dose, and in those graphs, the dose is in rads. Newer or more updated graphs, the dose would be in gray because you could convert if you wanted to, but it's the same concept. Dose is on the x-axis, surviving fraction is on the y-axis. On the y-axis, the graphs always begin at 100% survival. At this point where 0 dose has been delivered, they haven't been irradiated, okay? At 0 dose, everybody's surviving. But as dose increases, as you irradiate the population to higher and higher levels, the surviving fraction decreases. So does that make sense? That makes sense to everybody? The more dose that we give, the more cells will die.
Yeah, it makes perfect sense.
Yep.
Okay, because remember when we talked about factors that influence radiosensitivity, the main one was the quantity of radiation. The more radiation that you administer, the greater the biological damage. Same is true when we're thinking about cell survival. The higher the dose that's administered, the fewer cells will survive, or the more cells you will kill. But this is the basic setup for a cell survival curve, and it is simply a graphical representation taken from your data that tells you at which dose level what percentage of your population survives. Now, like I said, over time, there are many different cell types that have been studied. And what they have found is that there are generally two types of graphs or two types of curves or two types of responses, however you want to describe it, in terms of cell survival curves. And these are exponential and sigmoidal. So we're going to look at these and we'll talk about them today, and then we'll continue on with them over the next several lectures. But curve A is an exponential. It's essentially a straight line. Curve B is a sigmoidal. It starts, then has a curve, and then becomes a straight line. So these are the two types of cell survival curves. On the graphs that I gave you, some of them are exponential, some of them are sigmoidal, and you have a variety of each, and we'll be working with them over the next couple of weeks. So let's look first at the exponential curve. And the exponential curve, it's kind of an oxymoron, because when the exponential, or when cell survival of populations that exhibit exponential curves are graphed, they typically graph as a straight line. So here you see, this says on a linear scale and on a log scale, logarithmic scale. If you graph the exponential curve on a linear scale, it literally is a curve. And linear simply means the units on the scale are linear. So, you know, this distance is 333. Well, that same distance would still be 333, and then it would be 333. So equal distance means an equal amount. What you're most likely to see is exponential cell survival curves graphed on semi-logarithmic scales, which means the dose axis is linear, but the survival axis is logarithmic. In those cases, it graphs as a straight line. So you notice all the examples that I gave you in the handout are on a semi-logarithmic scale. So they are always graphing as a straight line for the examples that we're going to be using. So when we think about cell populations that follow or that graph as exponentials, their graph, like I said, on semi-logarithmic paper is linear. It will graph as a straight line. The slope will always be negative. So from its starting point at 100% survival, the slope is always negative or downward. What does that mean?
The survival rate's going down.
Survival rate's going down. The radiation is killing these cells, okay? If radiation, do y'all remember radiation hormesis? Y'all remember that theory of radiation hormesis effect, where radiation has beneficial effects? If you did a cell survival curve and radiation hormesis effect occurred, the slope might be flat, or there might not be a slope, or it might go up. Most of the time, that's not true. For a typical irradiation and typical cells, when we start to irradiate, the radiation will kill the cells. So the slope of the line will always be negative. It's going down. Higher the radiation dose, the more cells are killed and the fewer cells survive. The cell types that graph as exponential curves are typically what we would consider very simple cells or very simple organisms. So an amoeba, bacteria, those types of organisms. They're relatively simple organisms. They are easily killed by radiation. Hence, we see the slope of the line starts to decrease as soon as we start to apply dose. So cell killing begins immediately after radiation is applied. So these are populations that are simple and relatively easily killed by radiation. There's something that I want to illustrate, and to do that I had to use a linear graph. It was the only graph I could find to represent this concept. So you see the curve, the line is curved. That's because it's on a linear graph. For cell populations that exhibit as exponential curves, so that graph is exponential curves, equal dose gives an equal decrease in surviving fraction. So for example, we have 100% survival at 0 dose. 100 rads, or 1 gray, administered kills 50% of the population. Okay, 0.5, that's 50%. So at 100 rads, that's where our line is. So 50% of the population has been killed at a dose of 100 rads. In this population, every time you give 100 rads, it will kill 50% of the population that is alive. So 100 rads killed 50%. The next 100 rads kills 50% of what remained. So it's brought you to 25%. The next 100 rads kills 50% of the 25, bringing you to 12.5. Every time you you administer an equal dose, it kills an equal fraction of the population. So if we said here, if at 100% survival, there were 1,000 cells, after we administered 100 rads, there would only be 500. So it killed half of them. After we administered 200 rads, well, it cut the 500 down to 250. After we administered another 100 rads, it cuts the 250 down to 125. After the next 100 rads, the 125 goes to roughly 68, 67 and a half, and so forth. An equal dose of radiation kills an equal percentage of the population. With these types of cells, these simple cells that exhibit exponential cell survival curves. The relationship is considered all or nothing, meaning that one hit, and a hit, remember, is an unrepaired damage by radiation, to the area that controls reproduction, and the cell can no longer reproduce. These cells, again, are simple. They only have a single area, a single target controlling reproduction. And if we can inactivate or damage that single target, then we can kill the cell. And by killing the cell, again, we mean it can't reproduce. So for cell populations that exhibit exponential cell survival curves, what do we know? First, these are simple cells, bacteria cells, things like that. These cell populations only have a single target that controls their reproductive capacity. If that target is hit by radiation, it can be inactivated or damaged. If the target is damaged, the cell is killed, because killing, again, means it cannot reproduce. Cell populations that exhibit exponential cell survival curves are referred to as following a single hit, single target theory. A single hit can inactivate the target. They only have one target, so that's the single target. And if it is inactivated, the cells are killed and they cannot reproduce. So again, if we look at the graph, at 0 dose, there's 100% survival. As soon as you start to administer dose, the cell survival curve starts to go downward, a negative slope, and it is a straight line. Meaning that as soon as some small amount of radiation is administered, some cells are killed. The more radiation we administer, the more cells that are killed and the fewer cells that are surviving. Okay. So here's the question. That makes sense, or do y'all think I'm speaking Greek?
Greek.
Greek.
Definitely great.
That's what I'm afraid of. This is a little bit
Basically,
difficult unit to teach via Zoom. I was trying to figure out what to do. But
basically what you're saying is the most that we put in radiation, the more radiation we give somebody, the more chance that cells are going to die from that radiation.
Yes, that's true. But when we are looking at cell survival curves, don't think about an entire organism. What you are literally thinking about are specific cells that a scientist has grown and is studying. Because it's unethical to irradiate patients just to see if they die. So all of these are done as laboratory experiments with specific cell types. I don't remember which articles y'all chose exactly for your summaries, but some of them talked about cell survival curves. So this is really getting to that point. But these are laboratory experiments.
Yeah, I don't understand it either on the article.
Yeah, and then, that's why I said be careful of what article you pick, because you have to interpret it. So anyway, simple cells graph as exponentials, straight lines. They only have a single target controlling reproduction, If the radiation damages that area on the DNA, that target controlling reproduction, then they're dead. The cell may live out its regular lifetime, but when it comes time to divide, it will not divide, which means we've killed it. Okay? All right, so let's look at sigmoidal cell survival curves. Okay. So we said that when cell survival experiments are performed, cell types will graph as either exponentials or sigmoidals. So we know that curve number 1, because it's a straight line, and this is a semi-logarithmic scale, we know that this is the exponential. Exponentials are always straight lines. We also know that for every cell survival curve, the x-axis is dose, the y-axis is surviving fraction. So these are the things we've just said about exponential curves. Simple populations, a single target or a single area on the DNA controlling reproduction. If we can irradiate that target and damage it, that cell can no longer reproduce. So those simple cells follow the single hit, single target theory. One inactivating event on the only target they possess, and you kill the cell. So for these cell populations, as soon as we start irradiating, the curve illustrating survival starts to drop. It decreases because there's only a single target controlling reproduction. So now we're going to look at sigmoidal cell survival curves. So sigmoidal cell survival curves are those that graphically represent more complex cell types. So mammalian cells, our human cells, we are mammals, but the cells of more complex organisms graph as sigmoidal cell survival curves. A sigmoidal cell survival curve has 3 parts. Part A, shown here, because curve number 2 is the sigmoidal, is what we would refer to as just the slight negative slope. From 100% survival, it's decreasing, but it's not decreasing by much. So it's just sort of slowly going downward, very slightly downward. Part B, we see the curve turn. So it's got a sharp bend. That's called the inflection or the shoulder. Part C is a straight line portion, and it's called the exponential portion. So pretty simple. If you're an exponential curve, it's only a straight line. But if it's a sigmoidal, it's got this bend at the top and then the straight line. So the negative, slight negative slope, the inflection or the shoulder, and then the exponential or the straight line portion. So when you think about a sigmoidal cell survival curve, the first thing you will notice is that the slope of this curve is not constant. The slope of a straight line is constant. So when we look at curve 1, the exponential, the slope is always the same. Since the slope is the same, that's why we get an equal dose will kill an equal fraction. But with the sigmoidal, it's not. The slope is very shallow here, then the slope is changing here, and then the slope is steep here. But the slope is not constant. So what does that mean, or how do we interpret that? Well, if we look at this low dose range, A and really the first part of B, so the slight negative slope, and when we are getting to that shoulder, that turn, the slope here is very shallow, meaning it's not dropping very quickly. If we look at section C, the exponential, the slope here is very steep. What we can infer from that is that in the low dose range, this A, area of A in the first part of B, is that there's less cells being killed because the slope is going downward much more gradually. So in the low dose range, which is generically about up to 200 rads, we're not seeing as much cell killing as when we get to the high dose range. And the high dose range would be anything above 200 rads, where the slope increases dramatically. So what that infers is that these complex cells can accumulate damage. And when we think of that in terms of targets, what that means is that these complex cells have more than one target controlling reproduction. So for example, if we had a cell population that had 2 targets controlling reproduction, if we want that cell to be killed, if we want it to be unable to reproduce, we have to hit or inactivate both targets. If we just inactivate one, the cell is still able to reproduce. So these more complex cells have more targets controlling reproduction. So in the low dose range, they are accumulating damage, meaning you may be killing off some of their targets controlling reproduction, but probably not all of them at once. When we get to the shoulder, or the turning point on the curve, the shoulder is the point where maximum sublethal damage has been accumulated. You say, well, great, what does that mean? That means that you have inactivated or killed or hit, well, you have inactivated or hit all of the targets controlling reproduction except one. So when the curve gets to this shoulder, on average, you have killed all the targets but one for the cell population. So in the example that I used previously where I said these cells have 2 targets controlling reproduction. When you get to the shoulder, on average, 1 target has been killed in every cell. Then in the exponential portion, there's only 1 target remaining. And if it's inactivated, the cell is killed. But the shoulder is the point of maximum sublethal damage accumulation. What that means is that all targets controlling reproduction have been hit or inactivated except 1. And if we can inactivate that last one, then we can kill the cell. And again, by killing the cell, we mean the cell cannot reproduce. So, some conclusions for cell populations that graph as sigmoidal cell survival curves. The first thing that you need to know is that these are more complex cell populations. They have more than 1 target controlling reproduction. The second is that a single hit can inactivate a target. That's just the same as it was with the exponential cell populations. But if we want to kill these cells or cause them not to be able to reproduce, we have to inactivate all targets that control reproduction. So if you have a cell population that has 2 targets, you have to damage both of those target areas with radiation in order to kill the cell. If you have a cell population that has 5 targets controlling reproduction, you have to inactivate all 5 of those targets in order to kill the cell. Okay? So however many targets there are that control reproduction, you have to inactivate all of them. How do we inactivate them? By hitting them with radiation. And remember, a hit is irreparable damage. So these complex cell populations follow what we would call the multi-hit, multi-target theory. They have multiple targets, so we have to hit each target at least once to inactivate it. So it's going to take multiple hits to kill the cell. And again, by killing the cell, we mean that it cannot reproduce. So multi-hit, multi-target theory. Cells that graph as sigmoidal cell survival curves follow that theory. Now, since sigmoidal cells or cells that graph as sigmoidal cell survival curves have multiple targets, we need to be able to estimate the number of targets that they have. So we give that term a name and we call it the extrapolation number. And the symbol that we use is a lowercase n. So n is an estimation of the average number of targets per cell. So n is found by extending the exponential portion of a sigmoidal cell survival curve. So you find the straight line portion, you put your ruler in alignment with the straight line portion, and then you extend it out until it crosses the y-axis. Where it crosses the y-axis is your estimation of target number or your extrapolation number. So in this example, this particular cell population is estimated to have 3 targets controlling reproduction. So in order to kill a cell in this cell population, you would have to inactivate all 3 targets. But we find the extrapolation number by literally putting your ruler along the straight line portion and then extending that line out until it crosses the y-axis. Now, next Wednesday, we will start off. I'm not going to do any of it today because we're about to be out of time. But we will start off by determining extrapolation numbers on the curves that you have. Okay, so next, we won't use a ruler today, but we will need one next week. So extrapolation number, an estimation of the number of targets in the cell controlling reproduction. Now, another concept related to sigmoidal cell survival curves is the threshold dose or the quasi-threshold dose. Either term can be used. And it is abbreviated D sub Q. So quasi-threshold dose. Now, the quasi-threshold dose is trying to measure the width of the shoulder. Or another way of saying that is it's trying to estimate the dose that is causing maximum sublethal damage. Or another way of saying that is that this is the dose that will kill on average all of the targets except one. So we're trying to estimate a dose that will kill all the targets but one. And that's what we would associate with maximum sublethal damage. So to do that, it's pretty easy. Here we see our sigmoidal curve. Here we see the extrapolation line extended. And so for this population, it's estimated that there are approximately 10 targets. So that's already done for us. To determine D sub Q, I'm going to go to the next slide actually, because it tells us there. To determine D sub Q, after you have determined the target number, you have the extrapolation line. At 100% survival, which is typically 1 on your chart, you're going to draw a horizontal line straight across. Okay. Where the line intersects with the extrapolation line, you're going to drop a vertical line to the dose axis. And where that line crosses the dose axis, that is the threshold dose or the quasi-threshold dose. So if I estimated here, I may not be able to go straight down. I'm estimating that the threshold dose is a little bit more than 100 rads. So for this cell population, I know that it is a complex cell population because it grafts as a sigmoidal. I know from my extrapolation line that it has approximately 10 targets controlling reproduction. I know from my estimation of D sub Q that a little bit more than 100 rads will inactivate on average 9 targets in these cells. So does that make sense?
Yep.
One thing that I have a question about on the 10 is it has like a dotted line up there to that 10,
Uh-huh.
and the DQ is at 1.
Yeah.
So,
This dotted line is called the extrapolation line. That's what we use to estimate the number of targets.
so the dotted line would be the extrapolation line then.
Yes,
And where
this is the extrapolation line, that's what it's termed, to estimate target number. So this
and-
extends up from the exponential portion
Mhm.
is the extrapolation line. It tells us we have approximately 10 targets in this cell, per cell. D sub Q, when we're calculating D sub Q, we are going to use the extrapolation line. We draw a line horizontally at 100% survival, just straight across at 1. Where it intersects with that extrapolation line, we would then drop a line straight down to the dose axis. So we
Gotcha.
draw straight down to here, and wherever it crosses the dose axis, that is your threshold dose. And
Gotcha.
the threshold dose is the estimate, excuse me, is the estimation of the dose that's going to kill all the targets but 1. And in this population, since we said we estimated 10 targets, D sub Q kills 9 targets. So it has 1 target left. So the cell has survived. It still has reproductive capacity at that point because the cells still have 1 remaining target controlling reproduction. Did I answer your question?
Yep. I just wanted to make sure of where to look for which
Okay.
one and name them right. Thank you so much.
Okay, you're welcome. Any other questions? Any other questions?
Miss Dawn, where is this chart in the book? Is it the chapter?
These, let's see, let me see. There are it's in chapter 7
Mhm.
in the radiation safety book. Let me see if I can find a page number.
We're talking about the survival curve from
Yes.
mammalian cells,
Yes. It's
and
on
that's page
132.
133.
Yeah, 132, 133. We have not covered all that. The reality of it is there's not a lot of information in either of your textbooks relative to this material.
Mm.
Most of it you're going to have to get from class. You also could do, I mean, and there's some supplemental reading posted, but in either of your textbooks, there's not a lot of information posted.
Thank you.
All right. So we're going to stop here and we will pick up next week. We'll start out with a review and we're going to start out working on the curves that I have given you. Does everybody have the curves? Except I know Ivy doesn't have hers. I've got to get hers to her tomorrow, but does everybody else have the curves?